the most sophisticated computer in the world, with over 100 billion computing units, processing information as fast as 120 meters per second, weighing as little as 1.5 kgs, and using as little power as a single light bulb. Does such a computer really exist? Yes, it does, inside each one of us. The human brain is a remarkable specimen of nature's engineering, and glancing inside it has been science's biggest adventure. Now, a unique bank is ensuring that scientists across India can uncover its many mysteries. What happens inside this bank? That's a question of science. As far back as the 1st century AD, the great Indian physician Charak wrote in his Charak Samhita, "Thinking is an object for the mind." In the 16th century, French philosopher René Descartes echoed this notion in the famous statement, "I think, therefore I am." Both thinkers, set centuries and continents apart, recognized that higher consciousness and the ability to contemplate intelligently came from the mind. But they wondered, where in the human body was this mind? Anatomists, or students of human anatomy, were amongst the first to collect evidence. The earliest anatomists were ancient Egyptians. In the process of mummifying the dead, they got a good view of what lay beneath the skin. But they weren't always sure of what they were seeing. According to them, the heart was the seat of all intelligence. Legendary Greek thinker Aristotle agreed, believing that the brain was mostly useful for cooling the blood. However, one voice disagreed with the great philosopher, Aristotle's very own contemporary, Herophilus. Herophilus was one of the first Greek anatomists to dissect the human body. He claimed that it was in fact the brain and not the heart that was the seat of human consciousness. Over the next few centuries, different theories emerged to explain what the brain was useful for. Many, like the pioneering 2nd century anatomist Roman Galen, dissected animal cadavers for the answers. But it wasn't quite the same as studying the human brain, an act that was illegal in many cultures until as late as the 15th and 16th centuries. In the 16th century, a young Belgian anatomist named Andreas Vesalius published a detailed collection of anatomical drawings made from studying human cadavers. His pioneering work revived the practice of human dissection for scientific inquiry. In the 20th century, neurosurgeon Wilder Penfield made great strides in mapping different parts of the brain using different brain surgery techniques. Today, surgical study of anatomy is one of the key ways in which researchers continue to learn how the human body works. And it's invaluable when it comes to studying the human brain. If the human body is a finely synchronized orchestra, then the brain is its master conductor. It controls and coordinates every single function in the body, from how the heart beats and how the blood flows, to how our senses perceive and how organs react to stimuli. It controls our every movement and ensures our metabolism keeps us going. It determines how we learn and remember and is responsible for every thought, idea, and emotion we experience in the world. When it comes to understanding the brain's extraordinary abilities and functions, Bangalore's NIMHANS is one of the top institutes in India. NIMHANS, or the National Institute of Mental Health and Neurosciences, is also home to one of the most unique banks in India, the Brain Bank.
Initially in 1985, a brainstorming session in neurobiology was conducted at NIMHANS. And Professor P.N. Tandon, a eminent neurosurgeon from All India Institute of Medical Sciences, he along with all of us just mooted the idea that we need a brain bank so that we have access to brain tissue, so that the researchers can have an access to the human brain tissue instead of extrapolating the studies from the lower animals.
10 years after that, in 1995, the National Brain Bank was established. Today, the Brain Bank contains over 150 samples of human brain, each one donated for scientific research by survivors of the deceased. It's the only research resource bank of its kind in the country, and for the last 2 decades, it's performed a critical service to the scientific community. Whether it's a neuroscientist studying brain injuries, a medical researcher studying diseases like Parkinson's or Alzheimer's, or someone studying psychiatric diseases like schizophrenia, all they have to do is approach the National Brain Bank and ask for a brain sample absolutely free of charge. Diligently preserved samples at the Brain Bank are ready for them, each carrying important clues to unlock how the master orchestrator of the human body functions. Suspended in a pool of fluid in the skull is the human brain, a compact organ of immense intricacy. The largest part of the brain is the cerebrum, and it is here that much of our higher brain function occurs. It is divided into the right and left hemispheres. Researchers also divide each hemisphere into 4 parts or lobes. The frontal lobe is associated with reasoning, planning, parts of speech, movement, emotions, and problem solving. The parietal lobe is associated with movement, orientation, recognition, and perception of stimuli. The occipital lobe is associated with vision, and the temporal lobe is associated with memory, learning, and speech. The cerebrum or the cerebral cortex is full of cells called neurons that process information. The more neurons a brain has, the more complex its functions are. But how does the human brain accommodate over 100 billion neurons? The clue is in the ridged design of the cerebrum, and it's what makes us different from lower animals.
If you look at the lower animals, the brain is bald, it is smooth. But as we evolve, we start getting the convolutions. So in other words, we are increasing the surface area for more connections, more complicated connections, more understanding, planning, execution, all that. Look at the small brains like this duck brain. You notice it, this is smooth. But as we keep evolving it, we start getting the folds into the brain, and that's what you start finding it here.
Even the brain of an unborn fetus in the womb has more ridges than that of a lower animal. But these convolutions are just beginning.
The same things start becoming more complicated and evolves into the final human brain.
Beneath the cerebrum is the brain's limbic system. It consists of the thalamus, often referred to as the brain's switchboard. All information coming in from our senses or going out into other body parts passes through here. Below it is the hypothalamus, associated with emotional responses, our sleep cycle, and impulses of thirst and hunger. The limbic system also contains the amygdala, involved in memory and fear and how we process emotions. The hippocampus is involved with spatial recognition and converting short-term memory into long-term memory. In the back region or posterior of the brain is the cerebellum. Like the cerebrum, it is divided into 2 hemispheres. It's responsible for our balance and posture and helps coordinate movement. Finally, just below this entire structure is the brain stem. This is the part that is linked to heartbeat, blood pressure, and breathing. The stem consists of a midbrain, a pons, and the medulla oblongata that connects the brain stem and the spinal cord.
different parts of the brain have got a different compartmentalization and specialization. And understanding of this is very, very important for the clinician when they want to treat the patient.
Understanding the structure of the brain offers many answers to questions in neuroscience. But it's just one of the many approaches to studying it. The human brain is also studied biochemically to see how different chemicals, enzymes, and proteins affect brain function. Molecularly, to see how neural cells behave at the molecular level. And genetically, to see how our DNA and RNA are associated with the brain's complexity. But what exactly do these scientific studies have to do with people like you and I? We need them to treat and find cures for brain disorders. Today, mental health and neurology hospitals are filled with patients who suffer neurological ailments, as numerous as they are diverse. Be it brain injuries, lifestyle-related diseases, brain infections, or psychiatric and neurodegenerative disorders. A visit to the National Brain Bank's Brain Museum is a rare experience we could never get from a textbook.
If you look at this brain here, you see this area the brain is damaged while the other half is not damaged and is relatively normal. If you ask question why, immediately we know that this person was traveling without a helmet on the scooter. He fell down, met with an accident and went and hit the curb on one side of the road, and that caused a fracture of the skull and destruction of the brain. Normally we keep hearing about the stroke, especially the brain stroke, like a heart stroke. And the brain stroke, we have got two types. One is excessive rupture of the blood inside the brain because of rupture of a vessel, or the other alternative is blood supply is not coming. So when there is a rupture of the blood vessel, you start getting hemorrhage into the brain as you see it here. When you compare this with this brain is pale, but at the same time there is a damage here. This automatically tells me that the blood supply to that area is not coming because the blood vessel is blocked. So when you look at this here, this is a brain tumor with a cyst, multiple cysts. This is going to go slow, grow slowly, and there is cyst fluid, so I can resect it. But compare this with this here, there's a big mass here. It does not have a border, so I don't know where to cut and where to stop. And that is the problem with this type of a tumor. So this is slightly advanced stage than this one. This is a pyogenic meningitis. This can go on the surface of the brain and cause meningitis. This brain is taken from an individual who had schizophrenia and was in Nimhans Hospital for 17 years. But when I show this brain, I can show this as a normal brain. Just to tell me and remind me and everybody, in case of some of the psychiatric disorders, brain does not show structural changes but biochemical changes.
While the museum gives us a chance to understand the brain a bit better, it's away from the spotlight that the National Brain Bank's real mission is carried out. In these small rooms lie state-of-the-art storage and preservation units, configured at -80 degrees Celsius, the ideal temperature to maintain brain samples. Where do these samples come from? Each one has been painstakingly collected by the small but dedicated team of the Brain Bank. They're always on call in case the local hospital sends information about a recently deceased donor whose family has given informed consent to donate the brain. When this happens, the team is required to immediately conduct an autopsy and retrieve the brain within the next 4 to 12 hours.
We conduct the autopsy, and this whole procedure of taking out the brain normally takes 2 hours. And this can be done only in the mortuary and nowhere else. So, and there is a team of 4 people. We remove the brain immediately, bring it, and then we weigh it and take a photograph of it as a documentation. Subsequently, we slice the brain like a bread loaves.
At the Brain Bank, the brain is carefully dissected. The left hemisphere of the brain is preserved in formalin as a permanent record of its medical condition. The right hemisphere is sliced up into thin slivers. Each sliver is carefully prepared for storage in freezers.
and these slices are kept in the Ziploc bags, and they're all kept inside in a boxes, and these are all locked inside. And this freezer possibly contains about 100 brains.
Each bag is catalogued according to location in the brain and the disorder it has.
The different parts of the brain, we can see it, is labeled as cerebellum. We can identify the areas and take out these areas for research purposes. It's almost like a super bazaar, where on the racks we got different parts of the brain stored. We take it out and then give it to them, and they carry out the study.
The brain bank doesn't just keep samples of brain tissue. It's also a bank for other neurological tissues and fluids, most critically the cerebrospinal fluid. Cerebrospinal fluid, or CSF, is the fluid within which the brain is suspended in the skull. It is also found in various chambers or ventricles inside the cerebral cortex. It acts like a shock absorber for the brain and also assists in metabolic functions. Many brain disorders leave valuable clues in the CSF.
Whatever is happening in the brain is reflected in the fluid. So when I want to make a diagnosis, I can't take out the brain, but I can take out the fluid. So these are all the serum samples collected from a patient, and they're all labeled. So if somebody wants it, we'll just take out one vial and give it to them. And this contains about 5 ml.
The brain bank has got brain tissue preservation down to an art form, with each sample's biochemical integrity intact, be it its protein and enzymatic structure or its DNA and RNA.
That is a tribute to our freezers and to our staff, that the brain structure and the brain is well preserved for last 12 years, and it has not deteriorated, and it is useful for the study.
Even though the autopsied brain is no longer alive, thanks to the brain bank, it still contains valuable biochemical and structural signatures that can tell incredible stories. So the next question is, where do these samples go from here? What further studies are they used in? Samples usually end up here, in neuroscience labs conducting cutting-edge research.
Our laboratory mainly focuses on brain aging, brain degeneration and brain injury. The objectives will be twofold. One is looking at the mechanism of any event like aging or degeneration or injury. Second is see whether you can develop any drug.
This lab studies the kind of molecular and biochemical changes that happen when the human brain is damaged. Scientists study which parts of the brain are most impacted and the biochemical nature of this impact. Their learnings will help develop treatments to prevent further damage in the region.
When we get the brain tissue from the brain bank, we try to look for various proteins in that. So we mince the tissue in the beginning and then grind it up in a process called sonication, and then this is kept for centrifugation. So here we have the debris separated out from a clear solution which actually contains the protein solution. This is loaded onto a gel separated based on molecular weight. It is separated based on the size, and then this gel is stained, and after destaining we see the bands which represent various proteins.
In order to study the protein distribution for any damage to the brain, the lab takes 3 samples from the same brain. One sample is from the area of damage. The next one is from some distance away, and the final sample is from the farthest region from the damage. By conducting detailed comparative studies of these 3 regions, scientists hope to pick up precious biochemical information about the brain.
When we discussed with the project coordinator of the brain bank, we were able to get brain samples from different anatomical regions of the brain, starting from 1-year-old brain to 80-year-old brain. So we were able to compare across different ages, and we tried to do a comparative biochemical analysis of all tissues that were given to us.
By conducting such biochemical analyses of brain tissue samples, This lab has made important findings in how Parkinson's disease affects the brain. Parkinson's disease is caused when a chemical called dopamine is no longer produced in the brain due to the gradual death of the cells that make it. It's well known that this disease impacts a part of the brain called substantia nigra. But findings in this NIMHANS lab have shown that the disease also affects the frontal cortex, since it also shows reduced levels of dopamine. Such findings can help pharmacologists develop drugs that could treat patients and help manage this degenerative disease better. And this is just one discovery that would not have been easy to make without the Brain Bank. In the last 2 decades, the Brain Bank has facilitated research in a number of pioneering areas. It has helped shed more light on genetic disorders like genetic epilepsy. It has helped discover that the Indian strain of HIV is unique and different from that of the US and Europe. This discovery has prompted drug developers to create HIV treatments specifically for Indian patients. It has even led to the discovery of a special protein thought only to be present in the liver. Its discovery in the human brain led to a better understanding of how certain drugs are metabolized differently by different patients.
In that way, whatever experiments that we do in the lab, maybe using cells in a petri dish or by using a mouse brain, we have to ultimately extrapolate those findings to the human condition. So towards that, I think the Human Brain Bank has been extremely supportive and valuable for us.
But in spite of the unmatched role played by the Brain Bank, the NIMHANS facility is the only one of its kind in India. As compared to Europe and the US, who have between 20 to 40 such banks, the gap is tremendous.
The information about the Brain Bank, the utility of the Brain Bank, has not reached all the clinicians, all the pathologists, and all the researchers throughout the country. Ideally, there should be a Brain Bank in major research centers, hospitals, and clinical institutes.
But it's not just up to hospital administrators or doctors to launch a successful Brain Bank. The most crucial piece to the puzzle lies in the common folk like you and I. No organ bank can ever run smoothly without voluntary organ donation with informed consent.
If we don't donate the body, we can burn it or cremate it, it becomes ash or the dust. But on the other hand, if we give it, we can start understanding the basic structure of the body. If you notice it, how do we know the structure of the brain? How did it come to know of it? Because somebody donated it, they dissected it out and they saw the brain, then they said this is how the structure of the brain. This understanding comes only when you have access, and the access comes always when somebody donates it.
If resources like the Brain Bank become more common, more researchers around the country will be able to make exciting discoveries, bringing us closer to new age cures and treatments, and bringing us closer to understanding the magnificent organ that is the human brain.
We say, yes, in the Brain Bank, the tissue is readily available. Now you don't waste your time, you do your research.
If you'd like to share your feedback on today's program, please send your suggestions and comments to Vigyan Prasar, C24 Qutub Institutional Area, New Delhi 110016, or you can mail us at info@vigyanprasar.gov.in.